Good evening. Today we are going to see about the newer drug delivery systems, and which comes under our unit 4. And we are going to see about the introduction of the drug delivery systems, which we normally call it as a general method how the compound can be a pharmaceutical compound can be formulated, and it can be taken up into a body. And when you are going to take up these kind of compounds into our system, we should be ensuring that it should not have any kind of an side effects. And before getting up into this particular process, we need to understand that we have to understand the basic concepts. That is something which we call it as a top-down approach and a bottoms-up approach. Here, we are going to construct a nanomaterial which will be helping the drug to transfer into a body, which will be minimizing a lot of ill effects. So when we are going to construct this in any kind of synthesis process, we can able to understand that we will be able to have one basic component, that's the two basic components, which is what we call it as a top-down approach as well as the bottoms-up approach. And coming up to this particular process of a drug delivery systems, we will be able to understand that it is an approach, it is a formulation, and a technology, and the system for transporting the pharmaceutical components into the body, which will be needed with the safety achieved with this therapeutic effect. It means that without reducing the the drug effect, we have to ensure that this compound has to be effectively taken up into our system. And normally what happens here is we have a lot amount of methodologies. For example, we take medicine orally. We will be taking the drug in our intramuscular way, intravenous way, and we will be taking it as a topical applications, just like the headache bombs. And all these kind of processes been already existing. But still, why do we get into these kind of technologies, what we normally call it as a drug delivery systems, or if we call it as a targeted drug delivery systems? And here we can able to understand that there are many different kind of diseases present inside our system, which will be enhanced by, or when we are going to give a different kind of drugs, it will end up in a different kind of another disease conditions. So, and along with that, we will be understanding that there are then the normal conventional drug has got a higher dosage which we have to take it up, and it will be having a lower efficiency. And along with that, it will be also having a higher toxicity when we are coming under the assimilation as well as the elimination process. It means that when we have considering a particular drug, it should be when we take it up by an oral or an intramuscular or an intravenous way, the amount of the drug which is going to be transmitted or taken up to the target site will be comparatively very low. It means that there are a lot of eliminations which will be taking place from our oral cavity to the digestive system inside the stomach, and it will cross a lot amount of a barrier so that it can able to reach the target site. And in the meantime, the compound which is going to be taken up will be comparatively lesser, and at the same time it will be having some other side effects along with it. And what we are planning to do is, when we design a particular type of a compound, we will be understanding what is the composition of it, how the structure is being created, and along with that, we will be also knowing where and how. We call it as a route of administration, where we can able to understand that what is the nearest point where we are going to insert the particular drug so that it can be effectively reaching the target site. So depending upon the compound or the vehicles or the carriers which you're going to take it up, we have to decide the route of administration. And along with that, we will be also understanding what is the dosage required for that particular drug when it is been taken up into the systems. It means that when we are considering a nanoparticle and we are going to give it to the target site, it means that even in a picogram, it will be effective. We can always have a doubt. So when we are going to provide a particular drug, we will be always have a habit of taking a large and lot of drug, and we always imagine that whether the drug is enough or not. But still, what happens here is when you are going to take up these kind of drugs, we are going to give it to the exact point so that by and when we are understanding the route of administration as well as the dosage which has been required. Why? Because we are going to hit the target. It means that the compound of or the drug which has been taken up into our system, it will be having a very minimum concentration which will be enhancing that particular effect so that the disease can be controlled. Okay, and likewise the carrier and the cell interaction, it means that when we are going to design a particular drug, we and in the next few slides I'll show you different kind of carriers which have been used for the drug delivery system and how these kind of drugs are going to be interlinked between the carriers so that it can able to have an effective release into the target site. And along with that, we will be also understanding how the mechanisms, or what are the mechanisms which have been there, so that we can able to understand the cell, the drug delivery which has been given up into it. And when we are formulating it, and I already told you there are two basic approaches in case of nanotechnology. One is the top-down approach, another one is the bottoms-up approach. So when we are formulating a particular drug, we will be using either the different methods of the top-down approach or the bottoms-up approach. It means that every small molecules which will be interlinked to form a specific drug, which we normally call it as a bottoms up approach, or you take a bigger compound, break it up into smaller pieces so that we can end up with very fine particles, so which we call it as a top down approach. And we can use any kind of a methodology so that we can able to formulate that particular compound which will be having an effective kind of a reactions. And along with that, we have to understand what are the advantage of each and every different kind of compound. At the same time, we should be also understanding the limitation of that particular components. These are the pharmaceutical components. We will be understanding the pharmacokinetics as well as the pharmacodynamics of that particular drug. What is the time it is going to be retained inside our body? What is the effect of it, and what is the elimination procedures, and all these kind of process. When it is going to be understandable, we call it as an evaluation process. And what are the applications of it? And these are the things which we have to take under considerations when we are going to design a particular carrier for the drugs. And now what happens here is we are just going to call it as a targeted delivery or a sustainable release formulation. So we call this the next process as a targeted delivery as well as a controlled drug releases. It means that it will be having a high efficacy and it hits the target, which you call it as a site specific delivery. And at the meantime, what happens is it will be reducing the toxic side effect of it. It has got an increased conveyances, viable treatment for a previously incurable diseases, and the potency of a prophylactic applications and a better patient compliances. It means that, I'll take one point here. For example, when you're talking about a better patient compliances, means that when you are taking a huge amount of a drug, let us imagine that a small child of 12 years old or 5 years old, we give a paracetamol of 500 grams. It will be creating a big of confusions inside the kid. It will try to vomit and other things happens in the child. So what happens here is when we are trying to use a basic technology where we can able to release a minute concentration of the drug, even without the knowledge of the child, it will be having a happy face on the kid. It means that it will be having a better patient compliances. And beside all those kind of an emotional content, we have got an highest percentage of an efficacy which will be increased inside it, and we are specifically hitting the target, so that obviously the side effects or the toxicity of that particular compound will be comparatively very low. And in this particular session, we are going to see some basic technologies which have been used in nanotechnology. For example, the carrier-based drug delivery system, and we call it as a DDS here. Drug delivery system, in short, we call it as a DDS. And the carrier-based drug delivery system, transdermal drug delivery system, mucoadhesive DDS, and osmotic control DDS, and a lot of devices has been now available, and which you call it as a drug devices. Okay, so the drug delivery carriers. So when we are trying to design these kind of compounds, they are a special molecules or a system which encapsulates the drug. It means that it has got both an hydrophobic as well as an hydrophilic within the biocompatible polymers. Okay, we'll come back to one word out here. It's called as a biocompatible polymers. It means that when any kind of a compound, when it has been taken up into the system, there will be an antigenic reactions which will be happening inside the body. So what happens here is we have to send in any kind of a drug or any kind of a compound without creating much amount of an antigenic production or an antibody productions inside our system. Okay, so these compound without enhancing the issues, we normally call it as a biocompatibility. It means that it will enter into the body without altering much amount of an homeostasis, and it can able to have an enhanced effect inside the system without having any kind of an antigenic properties in it. And as we know that it is a targeted and a controlled release has to be there. It means that the amount of the drug which has been taken up into the system, it should be reaching the target, it should reach the specific target, and along with that, it should be having a controlled release. And at the same time, it should be biodegradable, non-immunogenic, biocompatible, and a high chemical stability. So it means that nowadays we always have a problem. Any kind of a compound which has been taken up into the system, it will try to adhere inside our body so that it can able to create an adverse effect. So what happens here is and we know that, right? The plastics have been, we have a stop on the plastics. Why? Because it is creating a lot of non-degradable issues inside the environment. Just like that, any kind of a chemicals which has been taken up into the body, that has to be degraded and it should be completely removed from the body. So this is what we normally call it as a biodegradability. At the same time, it should be having a non-immunogenic. I previously told you that when any kind of a component, when it has been entering into the system, it should not react or it should not induce an antibody production inside our body. So what happens here is, okay, there is always a question, right? So what happens if the antibody is going to be produced? So what happens there is the most of the drugs will be getting lost during the fight, and it will not reach the target also. And when the chemical is entering into the systems, we need to understand that these kind of chemicals will be passing up inside our body, and there will be a lot of chemical changes which will be present inside our system. It means that from the mouth, let's imagine that you're taking a drug inside our mouth, there will be amylase enzymes or a lot of enzymes which will be start breaking down on it, and when it passes into the intestinal tract, when it reaches the stomach, there is an acid present inside it which starts to degrade it, and there are a lot of possibilities that the chemical stability of this particular compound will be lost. Okay, and at the same time, there is we have to avoid the first pass metabolism and a minimal drug leakage during the transit. So I told you before itself, when we have a targeted drug delivery, the amount of the drug which has been lost during this particular process will be comparatively less. And so these are the basic points which has to be taken under considerations when we are considering the compounds which are being devised for taking up the drug. Okay, so here we have few carriers which have been listed out here. I'll just go through about all those things in a short while. Why? Because each and every topic is an engineering marvel. Okay, so it means that when we are talking about liposomes, there are thousands and thousands of papers which has been already released on it. And likewise the neozones, and basically what happens here is, I'll just give you a run through on all these things. So first one is called as micelles, liposomes, neozomes, ethosomes, transferosomes, cubosomes, virosomes, e-liposomes, cyclodextrin, gentrimers, nanoparticles. Then we have the nanoconjugates, microspheres, as well as resealed erythrocytes. And We get into each and everything in a very short while. Okay, so what happens here is the first one is called as an mishels, and previously in the class also we have been discussed that when we take a drop of oil, put it up into a water in a 10 ml of water, if you're going to mix it up, we will be able to get a compound which is called as an mishels. They are small compounds which are actually of lipids, and they will be and when you are mixing it vigorously, they will be making an into a smaller particles. And but what happens here is internally, if you are going to look into it, and here also we can able to see the picture. Internally we have got some amount of a space, and externally we can able to find the hydrophilic end and then hydrophobic tail inside the middle. Okay, so these kind of mishels, which we call it as a polymeric mishels, are formed from a self aggregation of an aggregation of an amphilic blocks, a graft copolymers with an hydrophobic part and an hydrophilic part inside it. So this is what it happens. And when we are talking about the nanotechnology, we always understand that there is one basic process which we normally call it as a self-assembly. So here, when we are going to see this particular picture, we can able to understand that this is the small kind of hydrophobic blocks which have been present here, and here also you can able to find the hydrophilic blocks which has been present here. And as it is a nanomaterial, it starts to assemble in a particular format so that it creates a specific structure like this. And the small black dots which have been present here is the drugs. So we can able to encapsulate our drug in this particular setup, and this will be an highly effective biocompatible materials. And there are many different kind of examples which are being, experiments have been carried out. So here you can able to load one kind of a compound or a multiple compounds inside it. And there are a lot of research which have been happening on it. Okay, and here also we can able to see that this is how the arrangement takes place, and this is a process where we call it as a self-assembly. And when we are talking about this, these compounds consist of several hundreds of the copolymers, and if the size of these molecules will be somewhere around 20 to 50 nanometers in its size. Okay, and there are different kind of the mishels which have been present. So one is what we call it as an hydrophobic interactions, and they are the normal kind of mishels which we use it. I told you, right, we can take a bit of an oil and we can just place it up in a water. We just shake it up vigorously. But when we do it in the experimental setup, we have to use a sonicator for that. Okay, so when you are just going to use a sonicator, we will be able to find that these particles will become very fine particles, so that the size of the nanomaterials will be reaching somewhere around 20 to 50 nanometers. And we will be also having an electrostatic interactions. I told you the first one is called as an hydrophobic interaction. The second one is called as an electrostatic interaction. The third one is called as a metal complications or an interactions, where we will be having a non-covalent materials which have been connected with the polymeric mishels. Clear? Okay, the next one, which is called as a liposomes. Here I have been taken one liposomes, and you can able to find that here also you will be finding an hydrophilic end and an hydrophobic tail. So previously it was been drawn as a small kind of a structure, but here it will, and again, right, when we are talking about a cell, we can able to find that it has been completely covered by these bilayer lipid molecules, and these molecules alone align together in such a way that it forms a particular smear kind of a structure, is what we call it as a liposomes. These are a spherical vesicle which has got an aqueous core and enclosed by one or more phospholipid or lipid bilipid layers. And here these are the one which has been first invented in the year 1961. And again, the name actually means that lipo means it is a fat and somes means a structure. So the liposomes again ranges from 20 to several millimeters. This forms a comparatively a bigger one. Why? Because of its self-assembly, and it is being completely filled with a non-lipid soluble drugs, and which will be retained until the liposomes will be getting destructed. And here we have can able to find few examples of the drugs which have been used, and a lot of drugs nowadays we are, I mean, the government, the FDA is approving it so that we can able to use liposomes for the treatment. And basic advantages, it is comparatively, when it is stable, it has got a very less toxicity, and it will protect the encapsulated drugs. And again, I told you before itself, when as it is a drug, we have to look into the pharmacokinetics as well as the pharmacodynamics of that particular drug. So it has got a better pharmacokinetics as well as a good therapeutic index. And it has also got a, hydrophilic as well as an hydrophobic drugs can be carried. And as an advantage of it, they are non-toxic, biodegradable, non-immunogenic, and less toxicity as told previously. And one of the major problem happens with us is it has comparatively and higher cost when there is a disadvantage of this liposomes. And the next one is what we call it as a neozomes. And why? Because again, right, in case of nanotechnology, the names will give you what we are looking into. It means that if it is a liposomes, we are talking about lipids. Okay, so likewise, neosomes, it is a non-ionic surfactants based structure. And if you're going to see everything, it looks somewhat similar. But the neosome are a non-ionic surfactant based vesicles which will be stabilized with or without the presence of cholesterol, or and along with that an anionic surfactant. And along with that, so if you are going to see this particular structure, okay, so here this is the skin, the epidermis. The dermis is present out here, where you can able to find the hair follicles which has been coming out here. But one of the advantage of this particular process is these neosomes can able to merge itself. It can able to convert itself into a smaller structures, and it can able to invade into the body. So can you able to understand that either these kind of structures will get into the body, and in some conditions it will breaks up and releases the drug, or it can be taken even to the dermis layer, where it can able to have that particular structure intact without losing much amount of the drug. Or we can able to make sure that these kind of drugs can be released normally wherever it has been passing through. So the amount of the drug which has been released, we can able to control it appropriately. And this is the structure of the neosomes. It has got a non-aqueous, non-ionic surfactant bilayers, and internally here we have got the aqueous compartment where the drugs will be stored up. Okay, and likewise a lot amount of drugs have been given out here. And here we can able to find that it is basically designed for the skin based deliveries. And basically, right, there are sexually transmitted diseases like an herpes. These kind of drugs have been applied onto it so that it can directly go and hit the particular microorganisms, whether it is a virus or a bacteria. We can able to devise these kind of techniques. And along with that, when we are talking about the insulin, most of the times these component has to be present inside our body for a longer time, so that it can able to be released slowly, so that the amount of the sugar which has been present inside our body can be maintained in an appropriate manner. So here the transdermal delivery of insulin can be also done using the neosomes. And the next one is called as an ethosomes. On the one end I have been made up ethosomes, and another one is called as a transferosomes. Ethosomes is again a combination of combination of ethanol along with the other components. Okay, so basically what happens here is they can be an hydrophilic, they can be lipophilic, they can be an amphiphilic. It can be combining having both a kind of a property, and it is having a higher flexibility because it will be having an addition of ethanol or surfactant. Okay, so previously in case of neosomes, we have been considering only about the surfactant, but in case of an ethanosomes, as it has got an ethanol present inside it, it will be having a much more flexibility. So this is how the evolution of each and every carriers have been happening. And when we are coming up to this particular structure, okay, so we can able to see that it has got a lipid layer, and an aqueous ethanol solution of the drug can be stored inside it. Okay, so as we can able to understand that acyclovir is again an antiviral drug which has been used to treat any kind of viral infections. And likewise, transferosomes, the name itself represents that these are the components which will be having an effectiveness and an elasticity and ultra deformatable vesicle, which is a drug carryable substance. And these kind of materials are basically used to transfer the drug from the or which we call it as a skin delivery method. Is it clear? And this picture will be also representing how these kind of components can able to pass through the skin surface without destroying its particular structures, and that's why these components are called as an transferosomes. Okay, so the ethanosomes as well as the transferosomes are some amount of the carriers which can able to have much more modifications than the previously told micelles as well as the liposomes. Okay, so the next one is called as a virosomes. So here, the previous structure has been bit modified with adding some different kind of an antigen molecules which have been present inside it. Sorry, the viral antigens have been added into it, and we already know that when a virus enter into our system or any kind of an host, it attaches only at a specific receptor. Okay, so here in case of a virosomes can be defined as a drug or a vaccine delivery mechanisms which contains an unilaminar phospholipid. And previously in all the pictures you can able to find a bilayered structure. This one is a unilayered structure, and it cooperates. It can able to accommodate different kind of antigens which are being of the viruses. And these viruses will be specifically attaching. We are talking about a targeted drug therapy. And so what happens there is these kind of the viral antigens will be attaching only to the specific receptors inside our bodies, so that at that particular point it will be injecting the particular drug so that the problem can be resolved. Okay, the viral membrane is being reconstituents so that it can able to form a virosomes. And these viral proteins are being conferred to the virosomes, which will be creating instability. And as we are taking only the specific structures of the virus, it does not cause any kind of infections. And normally what happens here is in the normal cases, we will be talking about the surface specific vaccines or component specific vaccines. Likewise, we will be taking only that the antigenic particles of the virus, so it will not cause any kind of disease conditions inside the system. And this will be having and again it is also proposed for an immunization methods. Okay, the cocklets, the next one which you're going to call upon it's comparatively a modified versions rather than why, because in the previous cases we have been seeing about something which is in a spherical structures, but this one is actually kind of a sheet. It's just a sheet, and this sheet is being rolled up, and inside it has been converted with it has been collected with a lot of different kind of components. And what happens here is when we are talking about these nanococcylates, these coccylates will be present, the drugs will be embedded just like a sandwich. These drugs will be present inside it so that the external surface can able to interact with the different kind of toxic materials which have been present inside our body, but nothing happens to it. But when it reaches the target, it can able to cause multiple diseases, or it can able to release this particular compound into that target site. Okay, so when you're going to see here, this is how it happens. Once it reaches the specific cell, okay, so it try to release the compound so that the drugs can be released into the organism. And maybe, right, this is the structure which we are going to say. It is a very old structure which you commonly call as a phagocytosis. And the phagocytosis happens when the compounds take up that particular compound into it. It converts into an endosome, and it reaches the liposomes, and finally the bacterial component. And why? Because this is very specific. Why? Because there are many different kind of bacteria which can able to survive inside the phagocytes. So normally what happens, there are a few examples, right? When you're talking about Mycobacterium species, or when you're talking about the Salmonella species, right? these components will be able to interact or present inside the phagocytes, and it will be taking it as a carrier so that it can travel in all around our system. Okay, so this is one kind of a technology which we call it as a cocklets, which is a sheet which is being embedded. Okay, the drugs have been embedded inside it, and where you can able to see that these layers are the where the drugs have been present inside it, and once it reaches, it opens up so that it can able to release the drug into it. Okay, and this is again a modified versions of it, which you call it as a cubosomes, when the structure is in a cubical structure. Okay, so when the structure, either it can be cubical, but when the arrangement of this structure, it will be forming something like a cubical structure, and it has got a similar kind of a property of a cocklets. But this, I mean, when along with that, what happens here is this is also a process where we can able to find it by using a top-down approach. So we take we create this. Okay, so this kind of structure so that the drugs can be embedded inside it and it can be delivered. And here, the liposomes we have been already seen. I told you that it is made up of a lipid material. And here we are introducing one more thing, which we call it as an E-liposomes. Why? Because when we are applying a sonication, so when the sound waves have been given onto it, it starts to release the drug. Okay, it means that we can able to understand that where the drug is being, where the target is being present, and we can send that particular compound appropriately to the specific place. And at that particular point, when we send in and the ultrasound, it can able to release the drug in a very specific combination. Okay. So the cyclodextrin is a sugar molecules which has been differentiated as an alpha, beta, and gamma. And the beta version is considered to be a more effective because it has got a larger cavity size, and many different drugs can be transferred into it. And here the next one is called as a dendrimers. Dendrimers is a nano-sized, highly branched macromolecules. It means that, okay, so when we are going to see this particular structure, we take a structure of methane, or you can take water. If it has got two hydrogen molecules and one oxygen molecule in the middle, it means that every structures it will be having an axis where the drugs can be holded inside it. So this one is comparatively a complicated structure which has got a central core, and at the same time it has got a lot of branches in it where these drugs can be attached along with it. Okay, so this is again a commonly used kind of a drug carriers, and this is called as a dendrimers. And now we get into the nanoparticles. Why? Because we have been already seen about different kind of nanoparticles in the previous classes. So the few of them are the dendrimers, the nano CNTs, the carbon nanotubes, the hydrogels, the polymer particles, the protein structures. All these things comes under the nanoparticles. And we already know that whenever a nanoparticles which is in a size of 20 to 200 is having nanometers, will be having a comparatively an effective results inside our system. And these nanomaterials provides different physiochemical properties because of its size, structure, and it's a high surface area, and it can able to absorb and encapsulate drug, and it can able to protect it. So these are the materials which have been commonly used as a potential drug delivery devices. And already we have been seen about the dendrimers, and the next one is what we are going to see about the nanopores. And here it is basically used for the cancer research and treatment. And okay, there are two different versions which have been given out here. This is a mechanical device which we normally call it as a MEMS or the NEMS, and this one is a naturally occurring structure where we can able to find that one strand of a DNA is being getting derived from the double-stranded structure. Okay, so here the amount there is a pore which is being present inside it through which the drugs can be also transferred. Is one the natural method where we can able to use these kind of structures, or we can able to devise a particular structure and where which we can able to have a reservoir, a semi-permeable membrane, and we will be also having an osmotic layer, the agents, everything present inside it, and through the smallest hole we can able to release that particular drug so that we can able to release the drug in an appropriate manner. Okay, The CNTs are considered previously, right? In the classes we already explained about the CNTs. So this is one kind of a compound where we can able to be get released or synthesized by the process of a bottoms up approach by using a methodology called as an electric arc deposition. Okay, so here when you are taking up the carbon molecules, it automatically does a self assembly, and it will be able to create a particular structure. Okay, it arranges itself as either the armchair model, the spiral model, or the zigzag model. It can be any kind of a model it can able to create, but it forms as a sheet and it converges it together so that it can able to form it as a tube. And when we have the buckyballs, which you're going to see it up later, we can able to create two semicircle structures into it so that we can able to protect it, or it will be acting as a capsule so that we can able to deliver the drug through this. And as it is been completely made up of carbon, it will not have any kind of ill effects inside our system. And the next one is what we call it as a quantum dots. The quantum dots are the one which are having an ability of releasing energy from it without taking any kind of energy from any other substances. It means that these kind of components, it's called as an cesium chloride, which will be able to liberate energy out of it. And these kind of compounds are basically designed for tagging it so that we can able to understand that where the problem or where this particular drug is being present. Okay, so the next one is called as a buckyball. I told you before itself. The buckyball and the carbon nanotubes both are having a similar kind of a property. Why? Because it is completely made up of carbon molecules. And what happens here is these are the structures which have been formed when the carbon molecules have been interacting in a specific ratio of an environmental condition. It means that it can be present in an inert condition, or it can be present in an activated or a reactive conditions, or in a vacuum. It reacts it appropriately so that it can form a buckyball, or it will be forming up a carbon nanotube. Okay, so here what happens here is these kind of drugs, components can be taken as such so that we can fill it up with any kind of components inside it, the drugs inside it, so that we can deliver it into the body. So as it is again made up of carbon molecules, it will not have any kind of an ill effects inside our system. And whenever we are going to tell that name, the nano shells, okay, so the nano shells are the one which will be made up of metals. Okay, so these spheres are being covered with gold or any kind of metal particles. So what happens here is whenever we are going to hear a term of a nano shell, it means that it is a material which has been covered with a metal, and majority it can be gold or it can be silver. Okay, so here we have got few pictures which have been given out here. Okay, so we can able to see that these kind of components can be targeted along with a nano material, and it can be taken up into the carbon, into the cancerous cells, where it can able to go, and when you're releasing an infrared, it can able to kill the cancer cells alone without killing the normal healthy cells. Okay, so this is how this is forming up. And now the next important topic, it's called as an immunoconjugates, and it means that we already have been studied about the antibodies. It means that antibody has got a structure which will be having the FAB as well as the FC sites. So FAB is the fragment of antigen binding, and FC is the fraction of crystallization. And here in this particular site, the FAB part, we can able to tag it up with any kind of it is very specific to the antigens, so that it can able to interact with the antigen. But this FC part is comparatively free, so that we can able to add a lot of different components into it, and where you can able to see that we can radio label it, we can add a cytokine, we can enable an immunotoxin. Okay, so we can able to do different kind of combinations in the FC part of it, so that with these kind of compounds which can be used for the drug delivery. And here if you're going to see it, This is a very good structure. You can able to find that the antibodies which have been present there, and the streptavidin has been also added along with it, and it is also tagged. Okay, so likewise, we can able to use the FC fragment of the antibody, okay, which can be used as a targeting agent. So this one is what we call it as an antibody drug conjugate. So we will be tagging up the antibody along with the drug in the FC part of it, and we send it up into the systems where which the specific antigens are being present. So we always have a problem with the normal cells and the cancerous cells, and there are different kind of markers of the antigens which have been present in the tumor cells, for which the monoclonal antibodies can be designed and or engineered. Okay, maybe we can use the term as engineered, and these antibodies which have been tagged with the drugs can be injected into the system so that it can go and interact to that particular site where the drug can show the effects only on the tumorous cells, not on any healthy cells. Okay, the next one is what we call it as the microspheres. There are two different kind of common components which have been present there. One is called as the protein or an synthetic polymers, and again this will be less than 200 micrometer in its size, and it has got a smaller size which will be coming up somewhere around 20 to 40 nanometers in structure. And there are different types of nanostructures or microspheres which we are not really getting up into it. There are a few of them are called as the muco adhesive microspheres. It means that it can able to adhere only on the mucous epithelial cell lines. And the floating microspheres have been basically designed so that whenever there is a blotting of an intestine or any kind of acids present inside the stomach, these kind of drug will not go ahead and attach along with the walls, it will be floating. And the magnetic, the microspheres, which already we have been seen about the the magnetotactic bacteria, which has got the MEM genes, as well as the magnetic particles which have been present, which is used for the tag and drag method. Likewise, this magnetic microsomes can be also used for the tag and drag method. And the polymeric microspheres, bioadhesive microspheres, and radioactive microspheres are also present. And here in this particular case itself, we will be also finding there are synthetic polymers as well as the natural polymers. The synthetic polymers, for example, we call it as a PMMA, and the degradable as well as the non-degradable materials is also present. In case of the natural polymers, we also know that the proteins, the carbohydrate, and chemically modified carbohydrates are all present there, which is basically used for which can be easily degraded. And when you're looking up into this, we have got large and large of basic methods of a preparation of these kind of components, where we can able to involve the solvent evaporation method, a single emulsion method, double emulsion method. Likewise, a lot of methods where we can able to use it to make these microspheres. And the next one is called as an resealed erythrocytes. We know that RBCs have been present, and it will be completely traveling inside our blood circulation. And we just engineer these kind of drugs, these kind of RBCs, which will be acting as a carrier. And there is a proved kind of a disease which we call it as an acute lymphoblastic leukemia, which we call it as an ALL. And this is an effective therapy for that. And these components are biodegradable, okay, biocompatible. It can travel a long distance throughout our blood circulation, and it can be entrapped. The entrapment of the drug is comparatively easy here, and the prolonged systemic activity of this drug can be present there, and many kind of activities have been happening along with this. And the next system, the carrier-based drug delivery system, is over. We are just looking into the transdermal drug delivery systems. It means that we are going to use patches for it. Okay, and I'll just try to give a small explanation about it. The transdermal drug delivery system means we can able, when a drug is been added as a patch over our body, the drug will be taken up through the skin, okay, which will be taken up into the blood vessels, and further on it takes up into our system. Okay, they are self-contained, and it will be also having a discrete dosage forms. And when it is applied on the surface of the skin, it will be delivering the drug on a controlled rate. Okay, so here what happens here is the avoidance of the first pass metabolism. I told you that whenever we take up the drug, there will be a lot amount of a loss, which will not happen in this case. And gastrointestinal incompatibility is also present inside it. It will not be present inside it. Why? Because it has been taken to the derm, into through the skin. And and the patients are not having any kind of a problem. Why? Because they are happy. They never know that this particular drug is being present or taken up into our system also. And finally, once the condition has been over, we can able to eliminate this one by peeling the patches off. And here you can able to find a kind of a drug which has been placed near to the ear, which we call it as the motion sickness. When we are traveling, people will be having a problem of vomiting. So these kind of patches which have been there, when you are just attaching it over the near to our neck, it will be able to solve that kind of problems. And the hydrogel transdermal patches, which has been basically designed for the treatment of burns. It is also a patch which has been attached over the surface of it. And the next one which you're going to see is about an insulin patch, where you can able to find small microneedles which have been present inside it, which will be injected into the system at a regulated speed whenever it has been required. Okay, so there is another kind of a process which you call it as a Sono Process. And here sono, in the sense, it means that the ultrasound. Okay, and we know that when you are sending this ultrasound over the surface of the skin, it will be enhancing the cell permeability. It means that somewhere around 20 kilohertz or 16 to around 16 megahertz of this the ultrasound is being passed through it. And here in the picture you can able to find that this is the dermis. Okay, and this is the epidermis and the dermis. And when the ultrasound is being given up over the surface of the skin, the molecules bit interacts a bit, it loosens down a bit so that the drug can effectively pass through it. And this method is called as a Sono Process. And likewise, the laser assisted transdermal drug delivery systems. And actually what happens is, this is the technology normally used for removing or permanent hair removal surgeries. Okay, so normally what happens it is in the facial hair, no, we will be they will go to the skin doctor, where they can able to send in the laser on the appropriate area where it burns down the root head formation, or it damages the little bit of a skin which will be recovered in a faster means. Likewise, here what we are trying to do here, instead of a sonar or an ultrasound, we will be using a laser which has been basically used along with the drugs which has been present over it. These laser will enhance the similar kind of a controlled disturbing of the cells, as well as the abrasion of the cell surface, that is the epidermis, so that the drug can enter inside it. So this is a common kind of a process which has been basically used for treating the skin or the facial scars. Okay, so and the next type is what we call it as the mucosal membranes. Okay, it means that it is a drug delivery systems. And here, this is the most commonly we will be able to find that some of the drugs are being taken, kept near to the gums or underneath the tongue, which you call it as a sublingual method. And here what happens here is, these kind of drugs will be attached over the surface of the mucus, which has been present inside the internal wall of the epithelial cell lines, where it can able to attach along with it, and then it hydrates, swells the polymer. There will be a drug release happens. You can find it up. The drug release happens. The hydrophilic macromolecules drugs will be taken up into it, and there will be an metabolism taking place, and the absorption happens. Okay, so here when we are talking about the mucosal membrane, it applies in the gums, so in the sublingual region underneath the tongue, or near to the gums. And many times, right, we know that we use inhalers in many cases. in case of wheezing, right? So in case of an asthmatic patients, they will be using an inhalers, and these inhalers will be having the drug which will be passed in a very high speed, and under pressure it will be reaching up to the specific level. Why? Because in case of an asthmatic people, the problem is because of the mucus which has been accumulated over the windpipe. So now what happens here is these drugs will be acting as a mucodilators, so that the effective process will be happening rather than taking a drug or an intravenous injections. And here on the eyes also these, and why? Because on the surface we will be having the micro injections which can also attached or like a lens. And along with that we also use a lot amount of the vaginal rings, the vaginal films and all the other parts. Why? Because it is also a complete layer which has been connected with the mucous membrane. Okay, so the next type is what we call it as an osmotic controlled drug delivery system. So here, what happens here is we will be having, and again, osmosis is a process whenever two different kind of conditions which has been present and a membrane which has been present there, from a higher concentration to a lower concentrations, the drugs will be moving on. Okay, so it can be taken up orally, or it can be used as a pumps. Okay, so what happens here is these kind of components will be okay. Here you can able to find a delivery orifice, and here there is a polymeric push compartment, and there is a semi-permeable membrane which are being present inside. So during operations, this will be moving up so that the compound will be released into the targeted site. Okay, so likewise we have got a different models which have been present here, and this is called as an implantable pumps which have been present on the arm, and which can be taken up. The drug will be moving up. Why? Because you can able to find that the salt components has been present, the drug components have been present, and the water chamber has been present. So when the water is been getting up into the chamber, the salt chamber will be moving up so that the drugs can be released outside. So this kind of implantable pumps is also present. Okay, so the next comes the drug devices. Basically, what happens here is we will be calling these ones as the stents. When there is an atherosclerosis, when there is an accumulation of fat molecules or the cholesterols inside the blood vessels, normally they will be using an angioblast or an angio. And when the angioblast has been done, the cholesterol molecules will be comparatively, it will be released outside. But what is the fate of that particular blood vessels? So why? Because when there is a clot which has been formed, or when the cholesterol starts to accumulate over it, the stability of that particular area, it will become dilated, it will become very loose. So what happens here is again it will lead for an accumulation of the cholesterol, or if there is any kind of a clot particles which have been present inside it, it also clots backs again, which creates a lot amount of an issues. So this one is normally called it as an coronary stents. Coronary stent, in the sense in the heart, the blood vessels which have been traveling around is called as the coronary systems. And when there is any kind of a problem happening in the surface of the heart, where the blood vessels have been traveling up, we have to remove that particular place, and we will be giving a bypass, or we will be using a stent. Okay, so here these stents are very minute particle, and it also has a possibilities of an inflating balloon which has been present inside. So this will release, expand at the area, and it gives us support. At the same time, it will be also release the drug which has been required in a very minute quantity, so that this further blockage will not be happening. Okay, so likewise here the next condition is called as a drug eluting micro systems, where we can able to implant it on the iris and corona, and where we can able to find that these kind of drugs will be releasing in the eyes. Okay, so the next one which you're going to see about the microchips, this is a very important kind of a device which has been happening up. And now what happens here is, before getting into this particular process, we need to understand what we mean by a NIMS as well as the MEMS. And the microchips or microchips are the smaller kind of devices which will be having an effective kind of an analysis process, and it also has got reservoirs which have been present inside it, and it can be manipulated, it can be activated by a wireless device, and all this lot of things have been present inside it. And this is a process which will be done by using the fabrication methods which we have been already discussed. And we will be seeing about the different methods which have been used here are the lithographic methods. By using the lithographic methods, we can able to design these kind of structures which will be acting as a microchips. Okay, so this will be doing a monitoring process. At the same time, it will be also doing a drug delivery as well as a therapeutic process. Okay, so the ionophores is the next method which we are going to discuss here. And here ion, in the sense we are talking about the electric current. Okay, so when these kind of compounds are being enhanced, like the sonar, like the lasers, these will be applying when an electric current is being applied over the surface of the skin. It will induce this kind of compounds, which why because they have been charged molecules, which will be acting as a medications. Okay, and the next one is what we called as an occlusert. It's an insert and an ocular, so it's called as an OCUSERT, insert and the ocular. So ocular, insert, occlusert. And these are a vinyl kind of a membranes which have been present, which has been highly, now it has been advisable for the glaucoma treatment. Okay, so and nowadays, right, we have got the PCA pump, and these are the things which have been basically used to in the children, where most the post-operative surgery, it will be a painful process. So these, the painkillers will be given in a very minute quantity so that the pain can be avoided. Okay, so the next thing which we are going to see is about the insulin delivery devices. So there are a lot of methods which have been used there. So one is called as an open loop delivery methods, where we use the Novopens, which we commonly inject the drug into the subcutaneous layers of it, or we will be also using some kind of an insulin jets which can able to pressurize through a small orifice with a high speed, which can penetrate into the underlaying tissues so that the drug can be present in the subcuticle region. So here, when we are going to see this particular structure, we can able to understand that these structures are being present due to the pressure. It penetrates into that particular area where it resides in the subcuticle region. Okay, so this is about the insulin delivery devices. A lot of methods have been there, and another one is called as a micropump insulin drug delivery systems, and the another one is called as this is again a computerized kind of a system where why because it can able to do the analysis how much amount of the blood sugar is being present there, and on the basis of it, it will be releasing this particular amount of insulin which has been required for that particular person. Okay, so normally in the other cases we will be injecting into the subcutaneous region so that it can retain for a longer period of time. Sometimes what happens here is there can be a condition, so when a higher concentration of an insulin has been taken up inside the body, sometimes it can end up in a very serious complications inside the patient. So that again we are devising a better okay, we are having a better system so that we can able to solve this particular process. Okay, so finally, right, these are the latest techniques which have been available now. What happens here is we have got the nanomaterial scale, the nano robots which have been present, which has been used for the diagnostics as well as the testing and other basic processes, and even the nano based surgeries have been also happening in this particular process. And as of now, we have been discussed something about the drug delivery systems. And in the coming up classes, we'll be discussing about how the cancer can be treated by using the nanotechnology. Okay, so if you have any doubts, just post it up. We will have a discussion later on. Thank you for your listening. Thank you. Have a great day.